It's a pleasure to be speaking here today. Thank you for the invitation. For the last 5 years, it's been a privilege for me to participate on the external review panels for both Systems X and now for PHRT, and to witness the growth of these exciting initiatives here in Switzerland. What I will do this morning is not to talk about my own work. But instead, what I'd like to do is to consider the question, what will it take to deliver the clinical impact of personalized medicine? And through this consideration, perhaps illuminate some challenges and opportunities that have not really been talked about so far. So let me begin by considering some of the issues that concern what we call personalized medicine. And I've highlighted in bold some of the things that personalized medicine means to me. It's the ability to develop what I call a new molecular taxonomy of disease based on the integration of both clinical and multidimensional omic data with a view to better tailoring management of individual patients. But let's not forget that taking things from a discovery phase to implementation in the clinic actually mandates the development of clinically viable biomarkers. And these considerations lead to an articulation of what I consider to be some of the major challenges required for delivering the clinical impact of personalized medicine. The first of those is, of course, how do we create this new taxonomy based on molecular information integrated with clinical information? How do we use that knowledge to develop clinically viable biomarkers to be able to stratify patients and individualize treatment? One thing that has not been talked about much, but I think is something for us to consider, if personalization is to be viable in areas like, for example, cancer, which I'll illustrate in a few minutes, we also need to be able to develop an enhanced repertoire of new drugs and clinical interventions. How are we going to do that? And finally, in the last few minutes, I'd like to consider perspectives which I think go beyond personalized medicine as an ability to treat patients, moving towards what I see as a coming revolution in medicine. I think for this audience, it's not necessary for me to emphasize the impact of genomics, proteomics, epigenomics, transcriptomics, all the omics that we've been talking about this morning. The availability of this information on a patient-by-patient basis has truly revolutionized our ability to create a molecular taxonomy of disease. This has largely been driven by the steep fall in the costs of sequencing human genomes. And as you know, in 2017, we are on the threshold of the $1,000 genome, and there's the promise that the cost will fall even further. We've spoken this morning about some of the issues connected with the collection and storage of this type of information, ranging from legal issues all the way through to physical storage. What I'd like to do in my talk is to use cancer genomics to illustrate some of the points that I wish to make. And the reason for this is that I hope you will agree that cancer genomics really has pioneered personalized medicine as we have come to understand it today. So this has started from about 2005 with the sequencing of the first cancer genomes to a point when today there are thousands of cancer genomes available. And moreover, we are beginning to see the first application of this genomic information in translation to the clinic, in particular, the stratification of patients for clinical trials using new therapeutic agents. But for those of you who don't work on cancer, I must emphasize particular issues that at once make cancer genomics both exceedingly rewarding, but also exceedingly challenging. And that is the idea that cancer is unique amongst human diseases as a process that is driven by somatic changes in cells. In other words, it's the accumulation of mutations in cancer cells that results in the manifestation of the disease. And what we know through the work of many laboratories is that the progression in tissues from early lesions to frank carcinomas is accompanied by a major shift in the genomic landscape with the accumulation of multiple mutations. Please bear in mind that these mutational processes give rise to noise as well as signal. And this is a point I'll come back to again. They also give rise to a multiplicity of different pathways that may actually move from mutation to the pathogenesis of disease. So I remind you of these points because I will allude to them in a minute. So in a way, what one might say is that the genomic landscape of cancer and the accumulation of mutations poses a challenge to a molecular taxonomy simply because it almost personalizes each individual case of cancer. And as you can see in this graphic, which illustrates the average number of non-synonymous mutations found across a range of common cancers, is that there are variations between different cancer types. Typically, cancers with an early onset, for example, in childhood, tend to have less mutations per genome than those that arise later. And finally, certain tumors associated with what we call mutator phenotypes can hugely increase the load of mutations in given tumors, which complicates, again, the issue of attributing a molecular taxonomy. And I think the nature and the scale of this issue is illustrated in this depiction. So what we're looking at here is a depiction of perhaps the 50 to 100 most common recurrent genomic alterations in a variety of different cancers, about 2,500 cases. And what I hope you can very clearly appreciate is that even for the most commonly recurrently mutated genes, there are relatively few cases in which the recurrence occurs. And moreover, in the way that Bert Vogelstein terms it, the genomic landscape is full of hills and little depressions rather than mountains or valleys. In other words, there are a number of genes which are likely to be important in the pathogenesis of cancer, but which simply do not occur at much above the predicted false positive detection risk. As we have heard this morning, one way to approach this problem, mutations are not very common, it's hard to attribute pathogenic significance because the signal sometimes does not rise above the noise, is to consider the way that cells are wired in biochemical pathways and networks, and explore the idea that some of these mutations may cluster in particular pathways, and therefore their meaning is somewhat larger than the meaning of an individual mutation. Such analyses have been done. This is work from the Broad Institute and Eric Lander's group, which, for example, has, again in about 2,500 cancer genomes, begun to attribute networks and finds about 15 to 20 different functional networks containing between 100 and 200 genes as being potentially functionally significant. So really, the point I want to convey to you is that even in a disease like cancer, which is very well characterized, relatively speaking, at the genomic level, we contend with many of these problems. In determining a molecular taxonomy and in connecting it to the mechanisms of pathogenesis of the disease. And this, in turn, you will realize, poses a number of challenges in actually taking this taxonomy and rising to the next challenge for personalized medicine. How does one translate this information to tailoring treatment for specific patients? Having left you with a moment to ponder what this means, Let me illustrate with an example another issue which I feel we ignore at our peril. And that is the idea that in the future, much of what we will come to learn about personalized treatment will come from data-driven research rather than hypothesis-driven mechanistic research. I don't agree. And I hope with this example, I will show you that at least in this example, it has been the confluence, on the one hand, of data-driven research coupled to years of hypothesis-driven research, which has actually led to clinical utility. And the example I want to cite is obviously something my lab has worked on for the last 10 to 15 years, which is high penetrance cancer predisposition associated with mutations in the so-called breast cancer genes BRCA1 and BRCA2. As many of you in the audience will know, about 5% of familial cases of breast, ovarian, and many other epithelial cancers exhibit a familial pattern of inheritance. In the case of breast cancer, we know that genes like BRCA1 and BRCA2, the so-called breast cancer genes, predispose to these types of cancers when inherited at a very high penetrance. What we also know, and please mark this because it becomes important in a minute, is that these are not just enlarging our knowledge of rare inherited familial syndromes, but in fact, similar changes are known to occur through somatic mutations which affect the majority of cancer cases. It's almost 20 years ago to the month when my laboratory discovered that the breast cancer 2 gene maintains the stability of human genomes through essential functions in the protection of chromosome structure during cell division. Work from my lab, as well as many other labs, over the years has now elucidated the mechanistic basis. And what we know, actually in atomic detail, and this is work from my lab from Jude and Yang and Kalina and Alessandro, is that BRCA2 functions in DNA repair to control an error-free process of homologous DNA recombination, which enables double-strand DNA breaks in the genome to be repaired effectively. And in its absence, you get more and more mutations accumulating, which is what predisposes to cancer. But the corollary of discovering the functions of BRCA2 in DNA repair has meant that there is the opportunity to ask, do patients who inherit these types of mutations respond better to DNA-damaging therapies? And this is work from Nick Wardell and her team, Sean Morrison and others in Australia, on about 250 pancreatic cancers. And I want to make a couple of points with this slide. This is just charting here the number of patients who respond to targeted DNA-damaging agents versus the genomic signatures that are characteristic of BRCA1 and BRCA2 mutations. And I hope you will easily see that patients who harbor those signatures respond exquisitely well to these DNA-damaging agents, as would have been predicted from hypothesis-driven mechanistic research. But to enlarge that point, you will also see that amongst these patients, those who actually have BRCA1 and BRCA2 mutations are in a minority. Many of these patients don't have mutations in BRCA1 or BRCA2, but they still have the signature that says that the biological processes controlled by BRCA1 and BRCA2 have gone awry. And this is the important insight that understanding mechanism can lead to hypotheses that move from the specific to the general. This type of research has now, with the identification of mutational signatures, enabled data-driven research, and this is from my colleague in Cambridge, Serena Nix-Zainal, who has developed a typical mutational profile that signifies BRCA-like tumors. And she calls it the HR detect, for homologous recombination detect biomarker. This is a biomarker which has the potential to be applicable across a wide number of patients. And in this paper, published just last year, you will see in the first set of patients how positivity for this marker, the HR detect marker, correlates extremely well with complete remission, complete responses to DNA-damaging and targeted agents. So you might think a victory for an approach which takes mechanistic work and combines it with data-driven work to enable clinical utility. But it illustrates another point that I want to emphasize, particularly in the context of Swiss precision medicine. And that is the challenge of scaling, particularly biomarkers for stratification, to the level when they are validated for clinical utility. Please take a look at this example. What I've charted for you here from Mahan and Tenenbaum is the millions of patients who would need to be tested to identify just 250 cases per year when there's a biomarker prevalence of between 1 and 30%. And this is against many different common and important cancers in which the mortality remains high, and we desperately need precision medicine approaches. But you will see here that if you assume stratification biomarker prevalences in the general population of, say, 1 to 5%, which I think is reasonable for many, the population registries that will be required to do accurate testing is extremely high, and certainly for many of these, well above the Swiss population. So as was discussed earlier, this may be a signal that at least for certain diseases, you will need to be thinking now about Europe-wide or wider international collaborations. I think we've already heard this morning that if we are to move from small examples like the one that I've given you to truly game-changing, paradigm-shifting implications of personalized medicine, we need far more data. And we need better data. We not only need, in the case of cancer, I believe, millions of cancer genomes, but we need deep linkage to specific clinical phenotypes and associated samples, like, for example, peripheral blood, plasma, and so on. Many schemes have been proposed to achieve this, but all of them have in common 2 or 3 facets. The first facet is the need for syndicated, quality-controlled, and anonymized patient data that spans pathology, molecular tests, treatments, and outcomes. This, you will see, immediately mandates, as we were discussing this morning, that care providers already need to be setting global standards, not just for treatment regimes, which in Switzerland, of course, and in many countries vary from hospital to hospital, physician to physician, but also be setting standards for structured observational data collection and storage, and we've heard about some of this in the morning. A second facet is that these data by themselves are insufficient without very powerful means to be able to couple innovation research and trigger rapid service delivery. In other words, we need to develop total ecosystems in which this type of information can be collected and, for the benefit of patients, immediately returned to the ecosystem. And I think with SPHN and other initiatives, Switzerland is an excellent place to do this. And indeed, in the United Kingdom, as was alluded to in the morning, Andrew Morris from the University of Edinburgh has been charged with leading the Health Data Research Initiative, HDR UK, which has the specific goal of attempting this across the United Kingdom. So I've spoken to you thus far about two of what I call the key challenges required to realize personal medicine. Let me turn briefly to this issue, which has not really been discussed yet. Personalized medicine, particularly in the case of cancer, where typically hundreds of different genes may be contributing to pathogenesis, means we need to enlarge the repertoire of available drugs and interventions. How are we going to do that? Now, as many of you will know, pharma companies have estimated the cost of taking new medicines to the clinic as somewhere between $1 and $2.6 billion US. I emphasize pharma companies have estimated this because many others have challenged this. Of course, it's in pharma companies' interest to say that medicines cost a lot because the price of R&D is high. And indeed, I have seen credible estimates ranging from $50 million to $200 million per medicine, which are orders of magnitude less than the pharma company estimate. So please bear this in mind. But nevertheless, I submit to you that the status quo is simply not an option for us. We have to do something about this problem, and we have to do it now. And I think a lot of this will actually come from solving, at the technological level, some of the main challenges to the rapid and efficient discovery of new drugs. So, as you know, you could take your favorite biological system and end up with a picture much like my favorite biological system, which is there are complex networks of biological molecules interacting to yield biological phenotypes. And what this means for us is that it's extremely hard in this complexity to pinpoint the best targets for the development of new medicines. To make matters worse, it's been estimated that there may be upwards of 100,000 potential new targets encoded in the human proteome. May I ask any of you to hazard a guess how many of them have been successfully drugged by the pharmaceutical industry? The best guess is of the order of between 3,000 and 10,000 out of potentially 100,000 targets. Moreover, when you do target atypical, novel, difficult targets, not enzymes, not kinases, how do you predict what's going to happen? For example, when you disrupt a key protein-protein interaction, will the characteristics of these new drugs actually be the same as enzyme inhibitors worked on ATP competition? So to begin to address this challenge, in my lab, over the last 7 years, we have attempted in Cambridge to establish an integrated set of platforms which systematically tackles each of these challenges. One, new ways to find what we think will be the best targets for new drugs. Second, combining structural biology and synthetic organic chemistry to be able to drug novel targets. And finally, new approaches in cellular imaging, hyperdimensional imaging microscopy, to reveal how these new drugs may work. Let me start with this, which is really the work of Alessandro Esposito, a talented biophysical colleague in my lab, who has essentially built what I consider to be very exciting new instruments, which now have the ability to detect spectral information, changes in anisotropy of emissions from a polarized instant beam, and fluorescence lifetime in the same instrument using a single smart detector that operates in real time. And the ability to capture essentially all of the photophysics of every photon coming through the sample is enabling us to unmix signals, both with reporters and without reporters, to truly try to understand how pathway flux leads to phenotypes. We have at the same time established an integrated infrastructure, an ecosystem, if you will, which brings together people in different disciplines to enable this. And what I would like to briefly talk about is some of the new genetic tools that we have developed to identify new targets. So, as you know, biological complexity is encoded at the level of DNA. And this is a linear set of information which encodes three-dimensional protein structure. Most methods, CRISPR-Cas9, for example, directly interrogate the linear sequence. They can identify targets, but they don't really give you very much information on how viable these are as new druggable targets. So we decided in the lab to see if we could directly break what I call the shape code. And to this end, we have developed a method which, obviously, forgive me for saying I find it exciting, but I would, of course. But what we have done essentially is to harness protein evolution and go backwards in evolution to extremophilic single-cell organisms which have considerable protein fold diversity, and harvest some of these to create libraries with very high complexity, which can then be used to directly interrogate the protein shape code. And the way we do this is to be able to use these shape libraries to undertake directly phenotypic screens. We're finding that at a relatively high efficiency, these probes, the shape probes, directly bind to macromolecules, proteins, DNA, RNA, that lead to biological phenotypes that we want to interrogate. And this immediately gives us information via crystallography or homology modeling about the druggability of these sites and establishes a template for rapid conversion to structured small molecule design. We've now done several examples where literally in between 6 to 12 months, we can go from a screen to the design of the first small molecule probes, not drugs, but at least they're a starting point for further discovery, and we hope that these types of new approaches will make drug discovery more rapid and more efficient at a large scale. Let me end by inviting you to go beyond what we've been talking about in terms of personalized medicine. We've been focusing on tailored treatment. But if you're like me, you will be dreaming of a future when we actually know before the disease occurs who is most at risk, and we have the ability to detect and intervene early. There is abundant evidence, particularly in the case of cancer, that early detection, early intervention, and prevention is effective, cheap, and very, very good for patients because it doesn't involve toxic therapies with major side effects. This, I think, is an area where Switzerland should be investing. Again, I have a vested interest, which I will declare, which is over the years, when I have directed the MRC Cancer Unit, we have decided not to be just another cancer institute, but actually to focus on developing methods for early intervention in cancer. What we wish to do is to develop the ability to detect cancer early, to risk stratify patients who have early lesions, and finally to develop new approaches for therapy and intervention. And to do this, we've assembled a hub of groups who study the confluence of genetic and environmental risk in cancer predisposition, study in detail molecular and cellular events during early steps in carcinogenesis, and use this information to develop innovative enabling technologies to take these advances through to the clinic. And I submit this to you for your consideration, that early intervention in chronic diseases is being enabled by the data that we're collecting, particularly if it's longitudinal. But I believe across the world, perhaps also in Switzerland, we are not thinking enough about the problem of early intervention. I could not end my talk without referring to the World Economic Forum's idea that we have now moved beyond the ability simply to gather information and to analyze it through to the advent of cyber-physical devices, like everybody's mobile phones, that actually interact with this information. And I would like to emphasize that in terms of device development for early intervention, the fourth industrial revolution has promise and major implications. This is a study which was published last year, and you can see that analysis using AI algorithms of images taken by phone have enabled the classification of benign versus malignant lesions with high accuracy. And just consider the fact that we will have over 6 billion mobile phones on this planet in the next few years. The point I want to leave you with is here too, we need more innovation, harnessing cyber-physical systems for early intervention, disease diagnosis, and potentially patient benefit. So I hope in this journey that I've taken you through, I've illuminated a few points which may be worthy of your attention, which have not really been discussed so far in the morning session. But again, I can't really end. I started my career as a physician working in primary practice in Indian villages, and I can't end without emphasizing that we live in a very unequal world insofar as health goes. And this is just a depiction of the expected healthy life expectancy. And you can see dark green, the developed countries, and in light green, developing countries, including India. And nothing for me illustrates this disparity better than this, which basically charts infectious disease mortality, not on the basis of population, but on the basis of mortality. Do we need personalized medicine to address these issues? I would argue that developments in personalized medicine of the kind that we have been talking about have the potential to transform the management of disease in poorer countries as well as developed ones. And I would beseech you in your work, both for the moral imperative of transmitting this type of technology, but also for the research imperative of enlarging your patient populations, and potentially the financial imperative of reaching a much larger market with new interventions for personalized health, that you must consider how your work can impact not just in Switzerland or in Europe, but across the world. And indeed, in richer countries, we may not be immune from some of the financial issues. This is an analysis about 10 years ago, published by Fojo and Grady at the National Cancer Institute in the United States. And you can see here that in non-targeted use of new drugs, which are very expensive to reflect the prices of high R&D, that there are questionable still improvements in overall survival. And a question we must ask even now is, what is the cost-benefit ratio for personalized medicine in diseases like cancer, and how can we recruit people from health economics to begin to help us to address whether these are worthy measures? So I'll end there and simply say I've given you a perspective, but one of my favorite sages, the American baseball player Yogi Berra, often said, it's tough to make predictions, especially about the future. And I'll leave you with that. Thank you for your attention.